Yes, all of you, please switch on your camera. Yeah, there is one more new student, uh, J, and yeah, there is one more new student, J. It is J, Naila. Something like this has been written. Please introduce yourself.
On the video, yeah.
Yeah, pardon.
Introduce.
Good evening, ma'am.
Yeah. What
Good
is your
evening.
name,
My
Bida?
name is Naila.
Yeah, pardon.
My name is Jumbe.
Okay. So now we will start our class. All of you, please switch on your camera. Okay. I'm audible to all of you very clearly. Any problem is there? Okay. So, I will be sharing screen. We have done few topics in last two classes. We will do a quick revision. I will ask few questions, those who have attended the class. No need to, hesitate while giving answers. Whatever you remember, please share. Okay? If you will revise with each and every class, it will be very much beneficial for you in your exams. So, because there is 1 week gap in between our classes, it's a long gap, and if you are not revising, then it can be a problem. So, in the classes, we have done about the life processes, differentiating between living and non-living. Okay, we had discussed about the nutrition, how we are getting the nutrition, what is the importance of the nutrition, and mode of nutrition, autotrophic and heterotrophic, but we have done in the detail only autotrophic nutrition. In the last class, we were done with the topic photosynthesis. Yeah, Aditya, Nilofer, yes, please describe me what we had, discussed or what you have written in your notes. I was dictating while teaching and expecting that you were noting down.
Ma'am, we wrote about autotrophic nutrition.
Yeah.
And the last thing we discussed is the chemical equation.
Okay. So can you define that equation?
So in that equation, it describes how the plant makes its own food, like taking the carbon dioxide and hydrogen and converting it into carbohydrates.
Okay,
By
very good. So, yeah. So what is the storage form of, glucose in case of plants? Anybody? Anybody can answer me? Storage form of,
Starch.
yes, very good. Starch. Okay. Plants, they store in the form of starch, their food. And human beings, what is the
glycogen,
store? Yes, very good. Glycogen. So the difference between the glycogen and glucose were clear to you, I hope. Who is going to tell me?
Ma'am, starch is stored in the
No,
plants.
not starch, glycogen and glucose.
Yes, ma'am. Ma'am, glucose is a form of energy in plants, but glycogen it's a starch that's stored as in humans. Ma'am, excessive
Okay.
starch that's stored in humans.
Okay, it's, thousands and lakhs of units of what we can say glucose only. Okay. Whenever our body need that glucose, glycogen molecule is broken down and one single unit of glucose is released, if you remember. So this equation is very much important. I have told you in that class also. This equation of photosynthesis, you should learn it. It can be asked in any form. Now, apart from that, the question also comes events of photosynthesis. So these are the three events of the photosynthesis: absorption of the light energy, conversion of light energy, and reduction. I have explained in the detail in my last class. So this is also very important. Now we will move to our next topic, and it's very good that all of you are revising whatever taught in the class. Okay, now there is one experiment, or we can say activity related to this. We will discuss this so that your concept can be clear. Yeah. So in this activity, what you are going to do, take a potted plant with variegated leaves, for example, money plant or crotons. Now what we need to do, keep the plant in a dark room for 3 days so that all the starch is being used up. We are doing this. You have to take a plant and you should keep it in the dark room. So what is going to happen while you are keeping it in the dark? What is going to
Base
happen?
starch.
Yeah, there will be no photosynthesis process then. Okay, in the dark, there will be no photosynthesis process and all the starch will be used up during these 3 days. Clear? All the starch will be used up during these 3 days. And plant is not going to do photosynthesis. Now keep the plant in sunlight for about 6 hours. After 3 days, you will be keeping the plant in sunlight for 6 hours. Now pluck a leaf from the plant and mark the green areas in it. And what next you are going to do? And trace them on a sheet of paper. Okay, you have to trace all the areas of that leaf on a sheet of paper. Now dip the leaf in boiling water for a few minutes. Now you are going to boil the leaf. After this, immerse in a beaker containing alcohol. Carefully place above the beaker in a water bath and heat till the alcohol begins to boil. Now what happens to the color of the leaf? So while you are boiling and you are, just immersing in it alcohol, what is going to happen? The photosynthetic areas will be chemically activated due to the alcohol. Now what happens to the color of the leaf? After that chemical immersion, or we can say this chemical activity, we need to dip in a dilute solution of iodine. Okay. After treating it with alcohol, you are going to dip, or you can say rinse off with iodine solution. Okay. What is the color of the iodine solution if you have done this activity in your school or in lower grades also? Can anybody tell me the color of the iodine?
Blue black.
blue, black, the same color as I am underlining with some, brownish. Okay, brownish shade is there, brown, dark brown. Blue color comes uh, with the
Alcohol.
starch. After its, combination with starch, iodine turns blue-black. What you are saying, okay? It is originally iodine is not blue-black color. Okay, clear?
Yes.
After reacting with starch, it turns into blue color. Otherwise, it's brown shade. Okay, brownish in color, iodine. So in this experiment, what you have done, first you have kept the leaves in the dark to avoid photosynthesis. You are keeping it for 3 days, so all the starch is being used up. Then you are chemically treating it with alcohol in boiling water, and then you are treating it with iodine solution because iodine tests the presence of starch. Okay, what iodine is doing? It is testing presence of starch, whether starch is present in any compound or not, that you can do with the help of iodine solution. Clear? If the iodine is present, what is going to happen? We are going to get blue color. Okay, clear? If the blue color is present, it means there is presence of what?
Starch.
Yes, very good. So this experiment shows the presence of starch in the given leaf or any other compound, you can say. The same test you can do with other things or other plants like leaf, and in some plants if stem is performing the photosynthesis process. In few plants stem is also performing the function of photosynthesis, stem part. In that also you can repeat the same experiment and you can get this blue black color. So where this starch was present in the leaf, the blue color is showing off. Okay, and it is depicting the presence of what? Starch. These areas which are green in color at the center, these are not having starch, that's why they are colorless. Okay, so this is a simple activity to show the presence of starch after photosynthesis, that plants are doing photosynthesis and they are storing it in the form of starch in the leaves. I hope this is clear to you. Sometimes some twisted activity based questions are being asked, that's why I've discussed this activity. After that, here you can see this diagram. Who is going to tell me about this diagram?
Ma'am, the opening and closing of the stomata for exchange of gases.
Yes, very good. So this is the diagram or pictorial view of stomata. So what are stomata? Who is going to tell me? Anybody?
A form of pore type structure beneath the leaf.
Yes, very good. I don't know whether you have done the experiment or not, but while you are doing the experiment of stomata, I think in the 11th class also it is, and 10th class also we show students, you can very much clearly see the structure of stomata. Okay, so write down some points about the stomata. So stomata, these are the tiny pores. Tiny. They are very tiny. small in structure. So these are the tiny pores at the surface of leaves. You have studied all this in lower classes, but still I am
Ma'am,
giving you.
stomata is present in the epidermis, right? Lower epidermis.
Yes, mostly it is present in the lower epidermis, and in few plants, exceptions are also there. It is also present on the upper epidermis. Okay, monocot leaves. In higher classes, you are going to study monocots and dicot plants. But till now, you have to, keep in your mind that it is on the lower surface of the leaf. Stomata, these are present on the lower surface of leaf. Why on the lower surface of leaf? Because the upper surface of the leaf is always in contact of sunlight. And if they are present on the upper surface, what is going to happen? Sunlight, all the time, evaporation of water is going to take place in the environment. So on the lower epidermis, you can say they are utilizing few sunlight for the evaporation of this water. So this is a kind of benefit for the plants if it is on the lower surface. Okay, so the singular is stoma. Okay, stoma. Singular, we say stoma, but they are very large in number. We never say stoma. You will not read this term anywhere, stoma, because always they are in abundance or in large number. So that's why plural is stomata. Okay. So green plants take carbon dioxide from air for photosynthesis. You know that? Okay. And the opening and closing of stomata, write down, opening and closing of stomata is controlled by which? By which the opening and closing of these stomata?
Ma'am, guard cells.
Yes, very good. Guard cells. So where are guard cells in this diagram? So these are the guard cells. These yellow structural cells, these are guard cells. Stomata is not any cellular structure. So it is the structure created by guard cells. Okay. This pore structure, this central structure caused by guard cells, opening and closing of the guard cells, we call that stomata. So stomata is just an opening. Clear to you? Any doubt?
Yes, ma'am.
Okay. This porous part in the center, that is stomata. Okay. Apart from that, exchange of gases, write down, exchange of gases also occurs across the surface of stems, roots, and leaves with the help of stomata. Okay. Exchange of gases across the surface of stem, roots, and leaves with the help of stomata. Clear? Now you can underline and you can note down also that when gases, or you can say the water is not needed by the plant, It can be lost through the stomata, okay? When there is no need, the guard cells are in flaccid state. Flaccid means not swollen, okay? This structure is swollen, like air is present inside. Balloon, like when we put in air inside the balloon, it is filled with air. Just like that, the guard cells here, they are swollen structures, and because of that, the central space is much, and we say it that stomata are open.
Ma'am, so the stomata closes when there's no need of water.
Yes, and no exchange of gases is taking place. In this case, there is, you can see minute opening kind of structure, but it is actually closed. No exchange of gases and water can take place in this flaccid state of guard cells. Okay?
Okay.
Yeah. So this is the open structure of stomata, and this is the closed structure of stomata. Very important for the plant, for the exchange of gases and exchange of water also. So how does exchange of gases taking place? When plant needs carbon dioxide from the environment, what is going to take place? What is happening?
Photosynthesis.
Yes, carbon dioxide is entering the chloroplast and doing photosynthesis. And what we have read in the last class, that after combining carbon dioxide and water during photosynthesis, glucose is prepared, and apart from this, oxygen is also prepared. Okay, so this oxygen is again released in the environment with the help of stomata. Okay, carbon dioxide is going inside the leaf and oxygen is coming out in the environment. Okay, clear to you? Yes,
ma'am.
beta, anybody is having any doubt in this concept, you can ask me. Clear? So this is how exchange of gases is taking place, and same way water. Okay, when plant needs water, it is going inside, and when evaporation is taking place, the same water is coming, same water molecule is coming out of this stomatal structure. So one more point is there. The guard cells swell when water flows in them, causing the stomata pore to open. What we have discussed in this figure, that when water is going in the guard cells, they are going to swell. Okay, swelling or swelled structure is going to take place, and this pore kind of structure is created. This pore kind of structure is created at the center, that we call stomatal pore and opening of stomata. When this pore structure is there, we say it is stomatal pore and stomata is open. Okay, clear? Similarly, the pore closes if the guard cells shrink or flaccid. When they shrink, this structure is going to take place, and it is the closing of stomata, because there is no center opening for the exchange. It is like a closed gate. The gate is closed and nobody can come inside and nobody can move outside. It is closed. No exchange of particles or molecules. Clear? I hope this is clear to you.
Small.
Yes.
Okay. Question is being asked from this also. That's why I'm discussing. Now there is one more activity. We will discuss it. 2 healthy potted plants, which are nearly same size. Keep them in dark room for 3 days. Now place each plant on separate glass plate. Place a watch glass. So I don't know whether you have seen in your labs or not. This is the watch glass. Okay, it is like a very small plate made up of glass. Okay, place a watch glass containing potassium hydroxide by the side of one of the plants. The potassium hydroxide is used to absorb carbon dioxide. Now cover both the plants with separate bell jars, okay, as shown in the figure. Now these are the bell jar. This structure is bell jar, obviously made up of glass. It is covering the whole plant. Okay. Now you are just covering and keeping the plant inside a closed chamber. Okay. With a glass plate that is filled with potassium hydroxide. Okay. Now, what you are going to do in this activity next, cover both the plants with separate bell jars. Use Vaseline to seal the bottom of the jars to the glass plate so that the setup is airtight. Okay, no exchange of air or gases now can take place. Okay, because you have covered and sealed both the jars. Now, keep the plants in sunlight for about 2 hours. Now, in one pot, you are keeping this watch glass containing potassium hydroxide, not in the another plant. Okay. B part, you don't have any potassium hydroxide. Keep in your mind. Okay. Now, pluck a leaf from each plant and check for the presence of starch in the above activity. Do both the leaves show the presence of the same amount of starch? Yes, yes,
Ma'am, I have a doubt, ma'am. So potassium hydroxide is used to absorb the carbon dioxide.
yes,
Okay,
yes,
ma'am. So in the first case, when they're absorbing, I think there won't be any carbon dioxide, right?
yes. So
Okay.
there
Okay.
will be no photosynthesis going to take place.
Okay.
Okay. So who is going to tell me something about the, result of this experiment? Can anybody have any guesses? No need to hesitate again. Whatever comes in your mind, just speak it out.
In the
What?
first case, there won't be any photosynthesis because there's no CO2. But in second,
Okay.
yes,
Yes, in second, there is no potassium hydroxide and
that's why,
there will
ma'am,
be
there
no,
will be.
yeah, there will be no outside, we can say, carbon dioxide inside the jars in both the cases. Okay, outside carbon dioxide. But in B part, some part of carbon dioxide can be there because there is no potassium hydroxide is there to absorb. Okay. So in which case of these experimental setup, A and B, you are going to have what is the question? Do you have the same amount of the starch or one is having more starch and other is having less?
A won't have
Will, yeah, B part that will be having,
starch.
yes, okay. So because exchange of gases in both the cases from the outside environment, it is not going to take place. But while you are putting this jar, few oxygen is trapped inside and carbon dioxide also. So B part can utilize that part to make more starch in comparison of A part. Okay, it can use that carbon dioxide for making starch. Clear? While you are putting this in the sunlight for 2 hours. So photosynthesis is going to take place in both the cases, but this A part will be deficient in carbon dioxide. So starch will produce, the starch which is produced is less in quantity in comparison to B part. Okay. So sometimes again questions can be asked in twisted form to check your knowledge or your concept, whether it is clear or not. In assertion questions also. Oh, assertion is in 11th and 12th class, sorry. In 10th class, there can be some activity-based questions to check your understanding of the concept. Okay. So here are few points which you can note down again. So sunlight is essential for photosynthesis. We can just, demonstrate with the help of these two activities. Apart from that, autotrophs meet their energy requirement. This we have studied. After that, other materials like, write down, nitrogen, phosphorus, iron, magnesium are taken up from the soil. And water is also taken up by the soil in, terrestrial plant. Clear? Water that is coming for photosynthesis from the soil, not from the environment directly. Okay? While it's raining or like that. Water is going down in the soil, and with the help of xylem tissues, the water is going up from the soil and plant is utilizing for the photosynthesis. Apart from this, nitrogen, phosphorus, iron, magnesium, they are also taken up from the soil. So nitrogen is an essential element that is used in synthesis of proteins in case of plants also. Okay. In 11th class, you will study that the building block of protein is nitrogen. When you are chemically drawing the structures of protein, nitrogen is playing very important role. Okay, these are also asked in some 1-mark questions. That's why I am explaining to you that nitrogen is the essential protein. Yes, beta, just wait for 1 minute. Yes, beta, now I am audible.
Yes, ma'am.
Okay. So you should know from where nitrogen and other compounds are coming from in the form of nitrates and nitrites. One more important point here that can be also asked, that compounds are being prepared by the bacteria from the atmospheric nitrogen. So sometimes question can be asked in this form, that the plant is utilizing nitrogen and in the form of nitrates and nitrites, and by which organism help it is being converted in the form of nitrates or nitrites. Atmospheric nitrogen is being converted into nitrates and nitrites. So answer will be bacteria. Bacteria in the soil is converting atmospheric nitrogen in nitrates and nitrites in case of plants. Again, in higher classes, it is, whole nitrogen
Mom, could you
cycle.
repeat that point again?
Yes, yes, yes. I am explaining myself also because it's a bit uh, atmospheric nitrogen. Plants are taking up the atmospheric nitrogen for the synthesis of proteins that we have discussed. That for the synthesis of proteins, plants they need nitrogen, and nitrogen is obviously coming from the environment or atmosphere. But in case of plants, this atmospheric nitrogen is accessible to the plants, you can say, with the help of bacteria in inorganic form, nitrates and nitrites. Now, who is preparing this nitrates and nitrites for the plant? Bacteria.
Bacteria.
Okay. Which atmospheric nitrogen? Okay. In which form? Nitrate and nitrites. Okay. For the synthesis of proteins. Clear?
Yes, ma'am.
I'm repeating again one more time, last time. Plants, they do need nitrogen for the synthesis of protein. Okay? Plants need nitrogen for the synthesis of protein, because while you are drawing the structure of protein, nitrogen is present always and playing a very important role. Nitrogen molecule is playing very important role in the structure of the proteins. Without nitrogen, proteins cannot be prepared by the plants. But to prepare this protein, atmospheric nitrogen is being converted into nitrates or nitrites by bacteria. Directly atmospheric nitrogen cannot be converted into nitrates or nitrites by plants. It cannot be converted, but it can be converted by the help of bacteria. Okay. For 10th class, you should be knowing this only. Otherwise, in 11th class, you will study the nitrogen cycle to explain how this atmospheric nitrogen is converted into nitrates and nitrates.
Mom, the bacteria is cyanobacteria, right?
Yeah,
Mom,
cyanobacteria also, and other bacteria also are also there. Clear?
Mom, also blue-green algae.
Yes, blue-green algae, Azospirillus, and other bacteria are also there. Clear? So these are helping to convert atmospheric nitrogen into nitrates and nitrites by the nitrogen cycle. Okay. So just one line question can be asked from this point. That's why I'm just explaining this to you, that how atmospheric nitrogen can be utilized by the plants by the help of which, okay, so bacterias. But if in the NCERT book name of the bacteria is not mentioned, so most probably it won't be asked from you. But if you know the answer, it's very good. You are smart enough. It is showing that you people are smart enough and you are doing your study, self-study also. Very good. Now, next topic is heterotrophic nutrition. Or till now, we have studied about the autotrophic nutrition, about the photosynthesis and the plants. Now coming to the next topic, that is heterotrophic nutrition. So what is heterotrophic nutrition?
Mom, the animals depend on the plants for food.
Only on plants. Yes, anybody else? Very good.
Ma'am, when the organisms depends on other organisms for their food.
Yes. So there are three types of heterotrophic nutrition. What are these? Anybody is having any idea? Saprotrophic, parasitic, and holozoic. You will study a few points about this also. So the form of nutrition depending on the type and availability of the food material and how it is obtained, we can categorize into three categories. Now you can see there are, some stationary sources like grass or mobile like deer, which can move. We say herbivores, carnivores, and omnivores. You have studied in the lower classes. All of you are having the idea of these, है ना? Herbivores, carnivores, and omnivores. And there are few examples of like bread, mold, yeast, and mushrooms. What are these? Yeast and mushrooms and fungi, which just take up their, food material from dead and decaying matter.
And saprotrophic.
Yes. Yes, saprotrophic, and the organisms are called saprophytes.
Phytes.
Okay, now write down the definition saprotrophic. Saprotrophic nutrition is the nutrition in which an organism obtain its food. Now, saprotrophic nutrition is the nutrition in which an organism obtain its food from dead and decaying. Means decay, or decaying means rotten. Okay, rotten. After dead body that is being converted into decaying part, that is rotten. So saprotrophic nutrition is the type of nutrition in which an organism takes its food from the dead and decaying matter, matter, matter of dead plants, dead animals, dead animals, and rotten bread, etc. Okay, rotten bread, etc. Bread, mold. Okay, we get mold in bread. All of you must have seen when the expiry date of bread crosses 2-3 days, some white color of structure is there that we call bread mold. Saprotrophic, this sapro itself means rotten. Okay, clear to you?
Yes,
Now, very good. Now, some other organisms, they derive their nutrition from plants or animals without killing them. So what type of nutrition is this?
recycling. Parasitic.
Yes, parasitic. Okay. So you can write down the definition yourself. Who is going to tell me the definition of parasitic nutrition?
Ma'am, it's when an organism takes their food from depending on a host.
Yes, beta, the definition is correct, but you cannot write down in this form in the exam. Okay, very good. Anybody else who want to try? So the definition is being written in the exam in this way, that the parasitic nutrition is the type of nutrition. I'm just telling you the definition. I will dictate also, in which an organism derive its food from the host without killing it. Okay, I will dictate, but before that, tell me what is this host means. Not the party host. We are hosting parties at house, not that host.
The organism.
Yes. What organism? Like in case of biology, we are having these two terms, parasite and host. Parasite is the organism which is obtaining food from the host. Okay, the parasite is living inside or outside the body of the host. Like in case of lice, lice is the example of humans. Okay, here lice. What is happening in that case? We humans are the host. Lice is not killing us. Okay, nobody is being killed by the lice. Maybe they are in thousands of number in the hairs of human. But what they do? They derive the nutrition from the host body, or we can say from the blood of human beings. So we are the host where this parasite is living. That part or that organism is called host. Clear? Parasite cannot live independently. It needs the body of another living organism, and that is called host. Clear to you? Now write down. Now write down. A parasite is an organism. A parasite is an organism. You can write down in bracket. The organism can be plant or animal. It can be plant or it can be animal. Which feeds on another, which feeds. Or it is like we humans generally use the term eating to get our food. It feeds on another living organism, feeds on another living organism. A parasite is an organism which feeds on another living organism called, and that is called, that other organism is called its host. Clear?
So,
Mom, we can also say organisms that derive nutrition from plants or animals without killing them, right?
What kind of example, Peter? Yes, we have said that this parasite is never going to kill host. Always it is deriving the nutrition without killing the host.
We can write
Okay?
that definition, right?
Yes, yes, yes. Either host will be in discomfort due to this parasite, or it will feel weakness in their body, Okay, like that, they are, causing some negative effect on the host, but they are not going to kill. Like in case of humans, this lice, if you know any of the person, it causes a very large level of discomfort. The person will not be able to sleep also in case of so many itching in the hair and scalp part. So this kind of discomfort or weakness can be created due to the, sucking of blood or deriving nutritional value from the host, which the host can utilize for its daily activities inside body. Okay?
Mom, so it takes the nutritions of the host.
Yes. It also always, doing some part of negative impact on host. That's why it's coming in the category of parasite. Okay. In again in higher classes, you will study different kind of associations between different organisms which are living in harmony. So they are not in the category of parasites. Or in case of parasites, always host will face some kind of negative issues like I have said discomfort or weakness or nutritional deficient like that. Okay. Parasite, it's not possible that parasite is living in the host and host is as it is healthy enough, you can say. Okay, health comes from where? Health come, you have studied in the ninth class. Health is wellness of mind and body. Mentally also, if a person is happy enough, satisfied enough, then it's in the category of healthy. So if the parasite is causing some kind of discomfort also in the body of the host, then it is in the category of parasitic nutrition. Okay, clear to you?
Yes, ma'am.
Yes,
Okay.
ma'am.
Apart from this, not clearly given in the NCRT, Holozoic nutrition. This is also asked few times. Holozoic, this term has been given in the NCRT, but not as such any definition or like that. Just write down the definition also. Holozoic nutrition. Few times it can be asked. It has been asked, sorry. Holozoic nutrition. What is holozoic nutrition? Or it can give the example and asks what kind of nutrition it is, parasitic, saprotrophic, or holozoic. So you must know what is holozoic means. Anybody?
Ma'am, it's when the organism depends on another form for their food. I mean, they take in the food and digest it inside their body.
Yes, to some extent you are correct, beta. Write down the definition. It is holozoic nutrition means heading will be holozoic nutrition. It means feeding on solid food, and the solid food can be plant or animal, plant or animal product. Okay, plants, it can be whole plant or it can be plant product, it can be animal or it can be animal product. Okay, and the process by which, write down, the process by which solid food, the process by which, sorry, animals take, we will write down like this, animals take the solid food into solid food into their body into their body by the process of ingestion. Animals take the solid food into their body by the process of ingestion. Okay? Clear?
Yes, ma'am.
Okay. Just write down further. Ingested food is digested. Ingested food is digested and absorbed Firstly, no need to write, I'm just explaining. First, the food is ingested, then it is digested, and now write down and continue the definition, and then absorbed. After digestion, it is absorbed by body cells of an organism. Okay? Of an organism. So example is humans, cattle, dog, deer, mostly higher organisms. They ingest their food. Okay? And this case of amoeba also, what we are going to study in our next class. That is also the part of examples, humans, cat, cattle, tiger, mostly higher organisms, giraffe, frog, fish, most of the higher organisms.
And amoeba also, right?
Yes, amoeba also. So sometimes the question can be asked, what kind of nutrition amoeba is having? What type of nutrition is this? By giving this diagram also. Explain the process. So first you will write down holozoic nutrition, then you will explain it. So this we will study in our next class. Till now we have studied about, in today's class, we have studied about the stomatal structure, activities related to the photosynthesis and sunlight importance, and then types of heterotrophic nutrition, parasitic, saprophytic, and holozoic. Okay? I have given you the notes. Please go through the notes. In the next class, I will ask a few questions before starting the class. Now, if anybody is having any doubt, you can ask me. Yes, anybody is having any doubt, you can ask.
No.
No? You have written all the points?
Yes.
I'm audible to all the students. There is no issue, I hope.
Ma'am.
Apart from that, while I'm dictating the notes, do you face any kind of difficulty?
No.
That's why I'm writing the main points also in between. Okay? No problem, no issues, no doubts.
No.
Okay, then please revise at home. I will give you the notes in the group also. Like last class I have given. Please do revise, okay? And whatever questions you are having in your mind, just write it down and ask in the next class. Okay? Okay, now, bye. Take care.
Thank you, ma'am.
Okay.
Bye,
Bye.
ma'am. Thank you.